Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. Today is June 1st, 2025. This video is on a JCI article titled Erythrocyte-derived extracellular vesicles induce endothelial dysfunction through arginase 1 and oxidative stress in type 2 diabetes. The accompanying editorial is titled Endothelial Dysfunction in Patients with Type 2 Diabetes. The truth is in the blood. The lead author is Sarah Constantino, and the senior author is Francisco Panini. Type 2 diabetes is associated with increased cardiovascular risk due to endothelial dysfunction. RBCs in type 2 diabetes patients can induce endothelial injury via upregulated arginase and oxidative stress. Extracellular vesicles are the key intercellular messengers, yet their role in type 2 diabetes-induced vascular injury remained unclear. This study investigates whether RBC-derived extracellular vesicles in type 2 diabetes impair endothelial function by transferring arginase 1 and inducing oxidative stress. RBCs were isolated from 68 type 2 diabetic patients and 35 healthy controls. Extracellular vesicles were collected via ultracentrifugation and membrane affinity methods. Extracellular vesicle uptake by endothelial cells was assessed via fluorescence imaging. The function was tested in mouse aortic rings. Arginase 1 expression and activity, reactive oxygen species markers, and RNA-seq were evaluated. Functional impact assessed using inhibitors and arginine 1 knockout models. What are the key findings? The key finding was in type 2 diabetics, RBC-derived extracellular vesicles were fewer in number but were taken up more readily by endothelial cells. These extracellular vesicles impaired endothelium-dependent relaxation via arginase 1 and oxidative stress, including NOx4 and reactive oxygen species. Inhibiting extracellular vesicular uptake or arginase activity attenuated the dysfunction. The limitation of the study was the ex vivo design. Patients had comorbidities and medications. Full in vivo relevance needs confirmation. Key takeaways: RBC-derived extracellular vesicles from type 2 diabetic patients contribute to endothelial dysfunction by delivering arginase 1 and inducing oxidative stress. Heparin and arginase inhibitors can mitigate these effects, highlighting therapeutic avenues. This study provides a mechanistic basis for RBC to endothelium signaling in type 2 diabetes vascular injury. Targeting extracellular vesicular pathways could reduce cardiovascular complications in type 2 diabetics. The accompanying editorial by Sarah Constantino and Francisco Panini said, RBC-derived extracellular vesicles were taken up by endothelial cells. These extracellular vesicles impaired endothelial function by transferring arginase 1, increasing oxidative stress, and reducing nitric oxide. The extracellular vesicular uptake, not quantity, determines damage. Heparan sulfate proteoglycans like syndecan-4 mediated the uptake. Heparin and arginase inhibitors block extracellular vesicular uptake and restore endothelial function, and these are the potential therapeutic pathways. The RBC-derived extracellular vesicles may also contribute to broader type 2 diabetes complications, these authors predicted, including hypercoagulability, myocardial infarction, and cognitive decline. Personalized strategies to block extracellular vesicular uptake may prevent vascular complications in type 2 diabetes. If you enjoyed this episode, share it with your colleagues. When we change how we see disease, we change how we shape lives. Until next time, stay curious, stay compassionate, and keep seeking wisdom. Podcast brought to you by Master MedFacts at mastermedfacts.com.